Hello everyone, our research proposal is on a 3D Alzheimer's brain organoid-on-chip model. First of all is the background and significance. Alzheimer's disease affects millions, yet we still struggle to understand its core mechanisms because the brain is incredibly complex. Current treatments only offer limited relief, partly because traditional models, like animal studies or 2D cell cultures, don't accurately represent human brain function or Alzheimer's pathology. National initiatives like the White House Brain Initiative highlight the need for innovative tools that can more realistically model neural systems. One promising approach is the use of 3D brain organoid-on-chip models, which combine the biological realism of organoids with the precise control of microfluidics. This technology allows us to simulate both healthy and Alzheimer's-affected environments in a more human-relevant way, improving how we study the disease and develop new therapies. Next is our objective and hypothesis. Our objective is to develop a 3D Alzheimer's brain organoid-on-chip system that can model both healthy and diseased neural tissue. This will give us a controllable, reproducible platform to observe Alzheimer's processes more accurately than current models allow. Our hypothesis is that integrating vascularized brain organoids with microfluidic perfusion will recreate key Alzheimer's features like amyloid plaques and tau tangles. If we can reliably generate these hallmarks, this system could become a valuable tool for studying disease mechanisms and testing new drug candidates in a more predictive and human-like environment.
Let me show you how our system works and why it's positioned to redefine Alzheimer's drug development. Starting on the left with iPSC selection and quality control, this step gives us something the industry currently lacks, which is scalable access to human-specific Alzheimer's models. We can generate organoids from healthy donors, familial mutation carriers, or even simulate sporadic AD, which is the form affecting 95% of patients. This means our platform is not only a model, but a modular disease library. Next, those stem cells undergo neural differentiation, forming cortical brain organoids, which are miniature, self-organized human brain structures. These organoids become the biological backbone of our platform, representing the closest in vitro approximation to the human brain that is currently available. At the same time, we will create a custom microfluidic chip that is the infrastructure that enables continuous nutrient and oxygen delivery, mimicking blood flow and preventing the failure points of traditional organoids. Endothelial or vascular cells will line the channels, giving us blood-brain barrier-like behavior. Then, we will embed the organoids and introduce microglia, which are the immune cells that drive neuroinflammation. With this step, we will achieve something that animal models and 2D systems cannot, which is real human, neuroimmune, and neurovascular interactions inside one integrated system. From here, we will initiate perfusion and long-term maturation, adding astrocytes to support network formation and inflammatory signaling. This will create a living, evolving Alzheimer's model that we can monitor continuously. After, through longitudinal monitoring, biosensors, MEA electrophysiology, imaging, and ELISA will allow us to track disease progression in real time. Instead of relying on endpoint snapshots, we can generate continuous datasets on amyloid accumulation, tau pathology, cytokine release, and neuronal decline. Finally, we will move into drug testing and reproducibility analysis. Because each chip runs under standardized flow, nutrient, and cell line conditions, our platform will offer something that the AD drug pipeline currently really needs, which is predictive, reproducible, human-relevant data at scale. So all in all, this workflow creates a one-of-a-kind solution for biotech and pharma, which is a vascularized, immune-competent Alzheimer's organoid on a chip system built for mechanistic discovery, therapeutic screening, and ultimately reducing the industry's catastrophic 95% failure rate.
Due to the complex nature of integrating organoids with our microfluidic device, we've identified several risks. The first risk is the interface stability. The organoid must attach securely to the device without disturbing fluid flow to allow for proper nutrient delivery and decrease mechanical stresses. Next, the perfusion of the fluid must remain constant. Endothelial line channels may vary in flow and structure, which can cause inaccurate vascular mimicry. Additionally, the long-term culture stability is important because the device requires cell differentiation. To maintain cell health and viability, it is important to ensure clear, sustained A-beta and tau signals. To address these challenges, we developed several alternative approaches. To improve device integration, we will adjust the chamber geometry and the organoid placement and initiate flow gradually to minimize mechanical stresses. To enhance perfusion reliability, we will modify the endothelial support conditions to strengthen the channel formation. To strengthen pathology induction, if our disease models A-beta or tau aren't clear, our induction conditions or combine genetic and environmental triggers.
The image shows a cortical organoid integrated within a microfluidic chip. Using this system, we can track Alzheimer's pathology in real time, including amyloid accumulation, tau hyperphosphorylation, and synaptic changes. The chip includes endothelial line channels